Welcome everyone to this first Monday Lunch Live of the year. I'm Anna Boltong and I'm the Associate Director at the VCCC. I'd like to welcome you. It's a really exciting special event we've got today for our first Monday Lunch Live of the year, it being World Cancer Day and us being able to celebrate consumers at the heart of everything we do with regards to cancer control. Before I start, I would like to acknowledge the traditional owners of the land that we're meeting on today, those of the Wurundjeri people of the Kulin Nation, and to pay my respects to elders past, present and future. The VCCC, for those of you who don't know, is a powerful alliance of 10 successful Victorian organisations committed to cancer control. The VCCC members and the Victorian Government are working together to position Victoria as an absolute trailblazer with regards to cancer education, research and clinical care. And we have a commitment within that to work with consumers, to engage them in all of the work that we do, to really advocate and facilitate a consumer-led and consumer-informed program of strategic work to improve patient outcomes ultimately. Today we're going to talk about and celebrate consumers in our work from a number of angles. And I'd like to talk about the broad definition of consumer that we use. And I know this isn't new to many of you in the room, but really the word consumer, and it's often debated, the way that we relate to that is to think about people affected by cancer in the broadest possible sense, from the angle of a patient, a survivor, a carer, a concerned friend and a family member. And as well as having world-class speakers here present to present to you today, we're also involving you by being here, you're not quite aware of it just yet, in a worldwide initiative because the theme of World Cancer Day is I Am, I Will. And at the end of the session today, you've all got cards there. We are going to ask you to write a message about who you are and what you'll do in the effort for cancer control, and we're going to take a group photo. So before I introduce our speakers, I want to share mine with you today, and in the definition that I just provided to you, I can also share that I am a consumer and I will advocate for optimal cancer care for all Victorians. So give you some time to think about what your message is today. Today you're all here to take part in and hear a wonderful story of patient-led cancer research that exemplifies the power and the importance of consumer activity in cancer research. Rare cancers is the theme today, and we know that it collectively causes more cancer deaths in Australia than any other single cancer type. Today we'll learn more about the NOMINATOR trial led by Prof Clare Scott, who is one of our absolute stars of research among the VCCC Alliance members. And that's an innovative patient-driven clinical trial exploring genomic profiling of rare cancers. The story will be told in 3 parts. The first will be Claire, who is a clinician-scientist from the Walter and Eliza Hall Institute. She's a medical oncologist at Peter Mac, the Royal Women's and the Royal Melbourne. Secondly, we're delighted to have in attendance Richard Vines, who's the CEO and founder of Rare Cancers Australia, and also Heather Camp, who is a consumer and research participant, highly valued in the study that Claire's going to talk about today. I've heard at least 2 of these speakers present before, and I know the extent of their passion, and so I've got a heavy job to time keep them today. And so what I've agreed with them is that when I stand up, that they've got 2 minutes to go, and when I get really close, they can feel me. That's when they need to stop, and we'll hand over to the next person. So bear with us and welcome to the podium, Claire.
Thank you very much. I'm absolutely honoured to be here today. So I'm beginning with my disclosures, but as you've heard, I need to keep to time. And I'm going to do that by telling this story in a very broad sense, but not sticking to any of the details that I have previously presented elsewhere. So I will define as I go through on the subsequent slides some of these terms that I'm just going to gloss over on these earlier slides. And in fact, the genesis of this story will actually be described to you by Richard Vines, So NOMINATOR is defined as the feasibility of performing genomic testing of rare cancers to match the cancer to treatment. And I've highlighted here amongst the co-authors Damien Key and Stephen Fox. So Damien is a medical oncologist here at Peter Mac, and as the medical oncology fellow on this, or medical oncologist on this program, he really ran this for me as the senior organiser. And Stephen Fox is the molecular pathologist here, and really nationwide the senior molecular pathologist who has the best comprehensive cancer panel for this, and it's the Peter Mac Comprehensive Cancer Panel that we used for this program. I want to highlight here and at the end that this research study was started by and for our patient advocates, for their patients, and this of course is none other than Kate and Richard Vines, who are the inaugural beginners of the Rare Cancer Australia's patient advocacy program. The premise is that next-generation sequencing analysis and immunoprofiling of tumour tissue from patients with a compelling rare cancer beyond evidence-based therapy can match the cancer to a molecular or immune type to guide selection of therapy with follow-up of patient outcome. And here we wanted to separate genomics analysis from treatment availability, because in this small study we would be unable to provide sufficient types of treatments to cover all the types of rare cancers, because there are way too many, and I'll show you that on another slide. So this was envisioned as a pilot study for 100 patients from around the country, and we thought that we would need a much larger study to go forward with. And at the time we started this, we didn't know what that study would be, but as I'll show you at the end, we now have the study to fold this into. So we were going to separate genomic testing from access to treatment. We wanted to assess the feasibility of performing genomic testing on matching cancers to treatment. This was a national multi-institutional observational study covering 5 states, 20 patients per state over 2 years with 100 patients, and these are the states and hospitals involved. So we wanted to have as our evaluation points the proportion of actionable mutations identified. We wanted to assess the availability and access to relevant drugs and trials, and the impact of molecular testing on patient care. Now, these are kind of rubbery endpoints, but they're still very reasonable endpoints. This then is what the study looks like. You have patients coming in with a compelling rare cancer diagnosis, and this was our definition of a compelling rare cancer diagnosis. So they had to have no identified dominant molecular driver mutation. So they wouldn't have, for example, a BRCA1 or 2 mutation, because we would roughly know what to do with those patients. They couldn't have any further adequate standard of care therapy available. They did need to have identified a poor outcome. They couldn't just have a rare cancer with a pretty good prognosis. And they needed to have the potential for benefit from therapy. They couldn't be terribly, terribly unwell. We then would take the clinical history obtained from their referring medical oncologists from around the country, and we would have had some input from their local molecular tumour board. We would then arrange for the genomic analysis for the Comprehensive Cancer Panel from our Peter Mac Comprehensive Cancer Panel, and then undertake a case review, and I'll show you what that looks like. We would then discuss their case with the result of the Comprehensive Cancer Panel in our variant tumour board, which takes place here on a roughly fortnightly basis at the Peter Mac. And then we would take follow-up from their clinical doctors and look at the impact on treatment outcome. We also collected blood for ctDNA and tumour tissue for immunoprofiling. We also had a questionnaire that the patient filled in on morbidity, psychomorbidity, and this is a validated questionnaire because we wanted to make sure that we weren't causing undue psychological harm to these patients. So this is a brief look at what the Comprehensive Cancer Panel looks like. It covers a wide range of cancer genes that are known to be involved in some way with cancer, and it covers not only mutations but other things like some RNA work looking at fusions, and to a certain extent other sequencing. But it is able to be changed as time goes by, and that's a good thing. So this is what one of our case reports looks like on the left-hand side of your screen. I'm not going to go into it in any detail, but it does cover what the clinical information was at the time the patient was referred, what information we received. We reach out nationally to find national experts on the very rare type of tumour that the patient has at the time, and over time, the information changes during the course of study, as it did for this case, and now the outcome for this case would be different than it was when this particular case was discussed, and we would go back to that case and inform them of the update. On the right hand of the screen, you can see the way the cancer report is presented to us by the Comprehensive Cancer Panel, and it goes on for 2 or 3 pages sometimes with references. So we then adhere to the MSK levels of evidence. I don't have time to go through these today because I would definitely run out of time allotted to me, but these are internationally accepted levels of evidence, and they go through from level 1 through to level 4. And level 1 is very unusual to reach, in fact impossible usually for the majority of rare cancers, although some rare cancers these days are reaching very high levels of evidence because they have very specific, very exciting findings in some ways, because we can have incredible matching of drug to a very specific driver of a very rare cancer type. So there is a lot of excitement and space for drug matching in rare cancers. But very often what we're doing is borrowing from another tumour type in the rare cancer space. So we need to do a lot of that, and we need to understand in great detail matching of aberrations to drugs. There's also the opportunity to understand when a drug is not well matched to a cancer. So there is a lot of detail to look at in the rare cancer space. But looking at exactly what we did here, we had accrued in October last year 69 of our 100 patients. We've now accrued 88 by last week. But if we just look at the 69 patients, you can see that the median age was 53, with a spread from 20 up to 76. And you can see that we really are accruing through all deciles of ages, including the 70+ range, evenly matched pretty much across genders, male and female. And then if you look at what we had done with the Comprehensive Cancer Panel at that stage, we had completed testing for 36 of the 100 patients, and that third are the third that I'm reporting on here. You can see that testing had failed for 8 patients, and the reasons for that only included inadequate tissue for 2 cases. For 2 cases, 2 were withdrawn, 1 because of disease progression and 1 because we chose to change that patient to whole genome sequencing, and that was because of delays in curation for the panel at that time. And there were still 23 patients who had to work their way through the panel. Now, this is a really interesting slide, and to me, this was the guts of what we really wanted. When talking about feasibility, this was really the very most important series of facts. And I really take my hat off to Damien, who curated all of this information. If you look at the large sectors of the pie chart, you can see that we're talking about soft tissue, the largest sector. So that's largely sarcoma, because in many ways in rare cancers, sarcoma is the best defined subset of rare cancer. They're very well organised, and they have a huge numbers of rare cancers within the sarcoma subset. Gynae is the other subset, which are very well organised. It's not just that that's my area, but 55% of gynae tumours are rare, and so gynae as a field are very well organised. And then you can see that upper GI, urology, endocrinology, head and neck, we know a lot about neuroendocrine, neurology, chest and skin. They all have large proportions of rare cancers within them. Those tumour streams have very well organised, very knowledgeable experts within those tumour streams. But if you look at the actual diagnoses, which we don't have time to go through today, they're a fascinating set of rare tumours. Now, a large proportion of these patients were referred from WA, South Australia and Queensland, where those patients have very little access to molecular testing, and that's why these cases came from those states. And what this means is those medical oncologists knew exactly who to refer to us, and that is the most exciting piece of information or feasibility that we gained from this study. So these patients are out there, and in Australia at large, those patients did not have any options for testing. So that's the first piece of information, that these patients were there to be referred. The next piece of information is, did we do them any good? But before I go on to that, I just want to make one other point. These little grids that you can see tell you about how rare these were. So the definition of rare is less than 6 per 100,000 incidents, and you get 4 in the grid for that. But if you get 1, 2 or 3 little grids in these boxes, that tells you that they were even more rare than rare, so going all the way down to super rare, which is less than 0.1 or 0.01 per 100,000. So these cases really are bordering on the super rare, or what we call less than 1 in a million. This is another grid, which I don't have a lot of time to go through, but I want to acknowledge Olga Kondrashova from my lab, a postdoc who's done a lot of this sort of molecular barcoding. This shows you along the bottom grids for gynae who are female, other subsets of tumours like sarcoma and so forth. And then up the top, this gridding by the degree of evidence. So many of them are grey, which is to say they're mid-level of evidence, and some have slightly more evidence than that. This shows you exactly what type of molecular aberration there was. So this is a very high degree of technical evidence and coding of the evidence. So again, we don't have time to go through it, but imagine how cool that's going to be when all 100 cases are coded. So we have done a lot of degree of analysis of this, and it is really fascinating. But this is the summary for you. It's much easier to take home. So overall, we have a couple of cases where we have very high levels, so level 1, 2A or 2B level of evidence, and I'm going to talk about some of those cases. We have the level 3A and 3B. These, particularly for 3A, we have, for example, cases where you have a mutation in a gene where we know something about that in other tumour types, but it's very hard to action it at the moment. And then we have a whole host of what we call level 4 types of genes. Now, these are genes where we don't have drugs today, but we're likely to have drugs soon. And that also tells us something about these rare tumours where right now we would know nothing about these cases for these patients at the moment. But this does tell us something about these cases, not only for their doctors in trying to understand their very rare cancers at the moment, but also for patients in the future with these types of cancers and where we should be heading for treating these cancers in the near future. And for a couple of these cases, we were able to make decisions about what not to treat them with. We were also able to change the diagnosis for some of these patients. So this is also very important, as I'll show you. So for 20% of these patients who had no treatment options and nothing more could be known about these cases, we were able to provide quite high-level evidence. So I'm just going to mention very briefly this patient because she spoke out and was very public in the national media. This is a patient who had this gorgeous photo of herself, both in the Sydney Morning Herald and The Age. So she has a rare endometrial carcinosarcoma, high-grade serous endometrial cancer. So she has a type of tumour which, once it's recurred, we have no recognised second-line therapy. And we found that she had a breast cancer gene, HER2, amplified very strongly to the point where she was then able to get HER2-targeted drugs free from Roche on a special access scheme. And she's been responding to those drugs really well. She, before she got access to those drugs, was really unwell at home, sleeping in bed all afternoon, not able to get out of bed much, and not able to look after her family at all. She's now taking her other members of family to the hospital most of the time and has really, had fabulous quality of life for many months. So that's a great outcome for her. This patient had a very rare sarcoma in the heart, and on biopsy, it really couldn't be identified much at all because it was a very poorly differentiated cancer on biopsy from the heart. But with the molecular evidence that we were able to provide, it looked like it really should have something to do with the skin. So once it was re-reviewed by the skin unit from Peter Mac and the pathologists from Peter Mac, it was actually identified as being a melanoma. And that meant that that patient had access to melanoma drugs from the PBS, so to double immunotherapy. And without being reviewed by the melanoma skin clinic from Peter Mac, that would never have happened. This patient comes from Perth. So that doctor was delighted and referred us his next patient, to NOMINATOR, I mean. So that also was an extraordinary outcome. We also had another case where, because of the Peter Mac panel, this patient was identified as having an MSI unstable tumour, and this was a pituitary adenoma, and that would not have happened otherwise. So just in summary, in my last 4 minutes, beating the 2-minute bell by 2 minutes, we have 50% of patients, or 53% of patients, where we had a level 4 or higher recommendation, all of which recommendations shed some sort of light on these patients' cancers. And all of these patients had rare cancers with no further light to be shed by treatment. So this really is encouraging. We have another 2/3 of the cases to be analysed, and we should complete our accrual on time. I just wanted to say that many of these cases are offered involvement in further research, a lot via the We Hire Stafford Fox Rare Cancer program, which I also run. And for quite a number of these patients, we do whole genome sequencing, which provides additional information. We have a shared collaboration with Sean Grimmond here at the VCCC. And for cancers, patients who have an aggressive cancer, which again has no other therapies available, we are finding additional information which can impact treatment. I also wanted to say that looking at patients' attitudes to testing as part of the iPredict program, JS-DES-I has an extensive research program looking at patients' attitudes to this type of testing. We certainly weren't able to look at that, but JS will be doing that. We have ongoing accrual. We were getting the right sort of referrals. I just wanted to talk about what's going to follow. So the Australian Genomic Cancer Medicine Centre, led by David Thomas, was funded by the current federal government to the tune of $55 million. And that is the sort of sized program you need to be able to link testing to treatment. So the Peter Mac will be the Victorian node for that, and there are 8 centres for that around Australia. So we look forward to that starting this year. And I think we've certainly shown rare cancers will be a large part of that, and it is feasible to include rare cancers in a nationwide program. So this was started by our patients, Kate and Richard Vines, and I really look forward to hearing from Richard now to hear how this study has affected Rare Cancers Australia as an entity and how we can continue to work together with him. So this is the, if you like, the platform of NOMINATOR if we look back at Australia and the many people who've been involved to make sure that this has been successful. And Diane Lindsay, formerly of NBCF fame, is our ethics and governance consultant. She's still based in Newcastle, but you'd never know it if you see how promptly she keeps us all in order. And I really have to thank her because of the very great debt we owe her to making sure that this nationwide study stays in order and on time. I'd also like to thank Heather personally because I'm not going to be speaking again today, and Heather's here to talk about it, and she's one of our patients. So a few more acknowledgements. Thank you.
Thank you, Claire, for encapsulating such a vast body of work in such a succinct amount of time. We are going to save questions until right at the end of the session. We think it'll be a really nice opportunity to invite all the speakers up at that time and be able to respond as a group or the area of expertise. So we'll welcome Richard next, please.
Thank you. I'm doing this without slides, so good morning, and you're in a PowerPoint-free zone. In terms of declarations or what have you, I'd just like to point out that Rare Cancers Australia will pretty much take money from anybody, and if you feel so inclined, it's rarecancers.org.au. So thank you, and a huge thanks to Claire for not only inviting us, but putting Nominator and bringing Nominator to life. And we worked very hard, and we were annoying, and we were persistent. And the reasons for that are long and complex, but fundamentally, we have a health system that does the greatest good for the greatest number, but it doesn't always respond either well or in a timely manner to people who have rare cancers or in some senses rare diseases. I remember the first time I met Grant MacArthur was at a conference or something in Adelaide, and I was very green, and I got introduced to Professor MacArthur, and I was suitably impressed. And he said, "So what do you want to do, take over the world?" And I looked at him a bit strangely, and he said, "Well, you must realise that as we learn more, every cancer will end up being rare. And hence, world domination is within my grasp. It's getting closer, but, you know, it'll all be for the best. Me and Donald will sort the world out." To understand what Nominator means to us is to understand that, you know, a couple of quite a good story and a bad story. I took a phone call several years ago, and I've never forgotten it, from a family, a daughter of a woman who had been diagnosed with Merkel cell carcinoma. And I don't know why she rang at the time she rang, because her mum was in palliative care. She'd had surgery and some radiation, and that was it. And she had been treated by, you know, a noted and respected oncologist, but not one who knew about a particular disease. A hard-working oncologist in the suburbs who didn't know that at that time people were being treated for Merkel cell carcinoma with pembrolizumab, or Keytruda as it's known. And we thought long and hard about how do we have an impact on that? You can't expect, we have 250 rare cancers on our website. How do we have an impact on building knowledge and understanding of those? And then we started to learn about the kind of work that Claire was doing and others were doing. And we thought, what if, in circumstances where there is no other hope, what if that tumour could be sequenced, analysed, and we could understand what was driving it and potentially look at treatments? A part of our work has been, for a long time, I say that we're an equal opportunity advocacy group. We advocate as hard to government as we do to the pharmaceutical industry. And if we know there's a drug and we know there's a patient in need, there is not much that gets in the way of us going after it, either through compassionate access or co-pay access or payment. We've raised over $3 million to fund cancer treatments in one way or another for patients over the last 4 years. But they're a bit like, we often tell the story of the little boy and the dad on the beach with the starfish, where there's 10,000 starfish stranded, and the little boy starts picking them up and throwing them back in the ocean. And the dad says, don't bother, son, you can't make a difference. So the little boy picks up the next starfish, throws it back in the ocean, and says, I made a difference to that one. And that's great, and that's a lot of what we do. We treat today's patients, and we don't treat them, we help them get the treatment they need. But the real secret in all of this is doing that and at the same time trying to raise the tide. Because if we can raise the tide, then we can save more, if not all. And the beauty of work like Nominator is that we have patients coming in from all over the country. We begin to understand what's driving cancer, and we help patients one patient at a time. We're still picking them up and throwing them off the beach, but we're building data and understanding and knowledge and science that then allows us to go to government and say, look, this is how you can treat these people. And the fact that one patient is suffering from a disease of 1 in 1 million or 1 in 10 million doesn't matter, because he's his disease is driven by this genetic disruptor or whatever the right term is, and we can treat them effectively. We've shown you that. So there's a new way for the government to deal with patients who need care rather than as it is at the moment, which is one cancer by location or histologically at a time. And it doesn't take a genius to work out if you run a pharmaceutical company and you've got a drug that works for 30 cancer types, probably you're going to start at the one with the biggest patient population and probably you're going to end at the one with the smallest patient population. And given the rate at which drugs are registered with the TGA and reimbursed through the PBAC, that's going to take a really long time. So this science, this incredible work that's been done and that is pushing into the future gives us the knowledge to go into the health system and say, no, there's a different way to look at cancer. And that will impact dramatically the way in which these people are treated. And that lady, next time, may have a different outcome. And we see those people every day, and now we've got somewhere to send them, which is wonderful. Because what happens, and the other story I wanted to tell you, what happens is if you deny patients any hope or any understanding or any knowledge, is they do crazy stuff. Because we went to a funeral 3 weeks ago of a 6-year-old girl whose parents had bankrupted themselves, trying to have her treated by a bunch of charlatans in Mexico. We paid to fly home a family recently from Mexico who had bankrupted themselves and could not bring their daughter home after she died because they were devoid of, they felt devoid of hope here, but they couldn't give up on trying to save their 5-year-old's life. And this changes that. It really does. It gives opportunity. Science and the technology and everything that you guys do changes that. And hope won't always win out, but hope with understanding will give people peace and allow them to stay within the system and not be exploited by some of those horrible people who sit over there. It's really, really phenomenal work. We're proud to be part of it in the small way that we are, and we will always continue to fight. And we've worked very hard, and I think things like NOMINATOR, trials like NOMINATOR and the work that's been done is best summarised by Greg Hunt, who's been a really constructive and good health minister. And he said when he was announcing the funding for the genomic cancer medicines program, he said, you guys weren't shrill, you didn't shout, you didn't bounce around, you just showed us what change would mean to patients, and we are very happy to support it. And that's the downstream power of what's happened here with NOMINATOR and the further downstream power of what will happen with the next phase of this. So thank you, Claire. Thanks to everybody who works in this place, because the need is great and the patients are deserving. Thank you.
Richard, thank you to you and Kate and all of your associates for helping so many starfish. Now I'd like to bring to the podium a lady that we like to call an expert by experience, for Heather to share what the study has meant to you in your journey. Thank you.
Thank you very much for inviting me to talk today. It was a total surprise, but I have been very lucky in my life to be in the right place at the right time for certain things. I'm 68 years old, and we had no sign of cancer or anything in our family. I lived in Doncaster on an orchard with my family, and growing up was a bit of a tomboy. And I had 2 sisters. I met my husband on the 3rd of November, 1973. A bit of a whirlwind romance. The second time I saw him, he proposed, and we got engaged on the 1st of December and married on the 23rd of February. My son was born on the 22nd of February, 2 years later. It's all right. And my second son was born on the 6th of June, 79. We had a very happy marriage. He was my soulmate. He loved his family and doing things that a family does. But unfortunately, in 1982, he contracted leukaemia. So we moved back to Doncaster East because he was being treated at the Austin at the time. And during that time, they tried lots of different drugs and things for him. They tried to get a bone marrow match, but unfortunately, nothing worked. So I even had to get people to come into the hospital to donate blood for him. Back then they just didn't have enough things on the side. But he got into remission, but then he died on the 27th of May, 84. So just a few days before my son's 5th birthday, and my other son was 7.5. So I built up a tremendous relationship with my sons. I love watching them play sport, and we're still really good friends. We speak to each other every day. So, but apart from family and friends, I have two other loves. The first one's the Melbourne Football Club, which I go when I can every week. I used to sponsor Shane Woden when he played for Melbourne. And my other love is Greece. And I've been 18 times. I've been to 36 islands, and I've followed, I love Alexander the Great, even though he's a bit of a devil. I followed him around where he was born, where his parents died. There's a few family shots. The grand dogs are in one of them, my son's two dogs. And in the top right-hand corner, top left is my grandson and granddaughter, and that's them also just below there. I took my son and his wife and the two kids to Greece because they hadn't been before, and so we had a wonderful holiday together there 2 years ago. When my husband died, I needed to have a goal, and my goal was to get to Greece. It took me 11 years to save, but I finally got there, and then my parents minded the kids. They were one was just got an apprenticeship with Mercedes-Benz, and the other one was a chef. So my parents kept an eye on them while I went to Greece, and they also loaned me some money at the end because I didn't quite have enough. So I travelled around thinking I was never, ever going to get back there again, so I fitted in as much as I could in that trip. Fell in love with the food, the people, the light, different kind of bright light over there. I can't describe it any other way. And at the end, I had 8 days in Italy, where I met my friend Margie, who's here with me today. So as I said, I've been there 18 times. My two big decisions of the day while I'm there are I look out in the morning and see which way the wind's blowing, so which beach do I go to? And the end of the day, it's which restaurant do I go to for dinner. So I have a wonderful time, a lot of swimming, walking, etc. In 2004, my son Troy moved to Queensland to live, and I moved into my apartment where I currently live. I went up to visit my son in Queensland in May. Mum had a stroke. She died in July, and my dad died in the October of the same year. So it was a bit of a heavy year, but anyway, we got through. So then in 2008, I started to feel a little strange round the neck. Went to the GP. He couldn't find anything. I had 4 different ultrasounds, still nothing. And finally, the cancer showed up. So Dr. Robert Miller removed my whole thyroid in March 2009. And then in 2012, I had a full hysterectomy using robotic surgery from Dr. Tom Manolitsis at Epworth in Box Hill. I was only in hospital overnight, which was amazing, and I went home the next day and I was walking around the court. No pain. It was just amazing. So after that, they gave me some chemotherapy, 3 weeks apart, and then I was given radiation every day for 6 weeks, and then another 2 lots of chemotherapy. Needless to say, I didn't get to Greece that year. But I was told that my cancer was rare and there was not a lot of treatment available. But I've been lucky that the surgery has been an option so many times. So most women or people, they don't have this same type of option. In 2013, I was feeling a little bit dizzy. So Dr. Alan Wise did an MRI on my head and discovered I had 2 small aneurysms just here. he suggested I see a neurosurgeon, and I saw Dr. Rong Jitu, who checked these every 3 months. And then he handballed me, sorry, football term, over to his colleague, Dr. Gautam Khurana, and we call him Dr. Vinny for short. In February 2016, I had a regular MRI check on the aneurysms, and he found I had a large carcinosarcoma on my brain up here, which he removed on the 15th of February 2016. I was awake during a lot of this operation, which was amazing. And then I had a few 10 days at rehab because I had lost my balance a bit. So they had me standing on Doncaster Road talking to me, but I also had to count all the white cars, trucks, bikes and everything that was going past at the same time. My cousins who are doctors were worried that this was a secondary and asked me to see my oncologist for a PET scan, and they were right. I had cancer on the lung as well. So I was given 6 treatments of chemotherapy, which at the start were reducing the cancer. And then I was told that it was inoperable. So I threw my watch away because I thought time doesn't matter anymore. I booked my family on a trip to Greece, and anyway, I was then going down to be measured up for radiation at Peter Mac at Moorabbin, and Dr. Pearlie Kaur was my doctor, and she said to me, "Heather, have you got a minute?" And I said, "Yeah, why?" And she said, "Oh, look, I've just been looking at your scan, but I want to get a second opinion." I said, "Yeah, that's fine." So 2.5 hours later, she came back to me and she said, "People have been looking at your scan. We think we can operate." And I was just like, right place, right time again. So I was sent to see Dr. Associate Professor Gavin Wright, and he operated on me on the 4th of March 2017. And his last comment to me was, "Now, Heather, I don't want you to take this the wrong way, but I hope I never see you again." So I was well enough to take my family to Greece at the end of May. We stayed for a month and we had a wonderful time. And when I got back from Greece, I had a PET scan and got the all clear, so I was very happy. In September and October, I had cataracts removed. And then I had a PET scan the following week I came to see the amazing Dr. Anne Hamilton, and they were all clear. I went up to the loft to have a coffee to tell my sons the good news, and I received a call from Dr. Clare Scott saying, "If you're still in the building, could you please come down and see me?" So I found her, and she told me about the NOMINATOR study, which I signed up for. What an amazing opportunity. I consented to further research, and this allowed them to test my cancer with whole genome sequencing. I told her about an operation I was about to have because I'd had another tumour form here. And so Dr. Clare organised, this blew me away, for somebody from Peter Mac to go over to Epworth where I was having the operation and collect part of the tumour and brought it back here, and they started testing it. So I just couldn't absolutely believe it. Amazing. So I was awake during that operation as well, and I'm even on YouTube for about 3 minutes showing a picture of the operation. And so I thought that was it, but unfortunately it came back again the same year. So I had that on the 26th of March. I had the next one operated on in September, and then when this one didn't want to leave me, it was quite a big one, and I had to have a blood transfusion. So again, Dr. Clare sent somebody over and took a sample, brought it back to work on. And then there was a hole left there, so I had to have another operation. So 3 operations on my head last year to fix up the hole. And earlier today, I had some radiation in December, and I've still got some cancer there in the bone. So this morning I went down and got measured up for another mask, had a blood test done, and I start radiation either later next week or early next week. And after that, I'm going on to the NOMINATOR program, and hopefully we can find a cure for my rare cancer. So thank you very much.
Heather, thank you. What resilience to go through all of that and to be here and share it with us so positively today. I did want to also speak to the power of our own VCCC Consumer Advisory Committee, which is a group here that is highly engaged. It reports directly to our board and therefore at the highest level has input into our strategic direction. We've got members of the committee here today, and I have over my career had a lot of exposure to different consumer advisory committees, but I'm delighted to say that really the way that this group works together is so impressive, and it feels like a real exemplar in the sector. So Katya Gray, who's representing the committee today at very late notice, and we need to be very grateful for that, in place of Sophia Than, our chair, who couldn't be here at the last minute, is just going to share with you a little bit about the achievements of the committee over the last year. It's been a big year of work, and talk a little bit about moving forward before we invite questions from the floor to all of our panel members today. Thanks, Katya.
Thanks, Anna. My name's Katya. I'm a member of the VCCC Cancer Consumer Advisory Panel, and I had the honour of standing in for the chair, Sophia Than, today. My World Cancer Day declaration is I am the Consumer Coordinator for the Walter and Eliza Hall Institute, and I will continue to enable consumers to work with researchers to make better research outcomes. Thank you to the speakers before me. It's wonderful to see the demonstration of a researcher and a consumer working hand in hand, and hearing the consumer voice to make the program better. The Cancer Consumer Advisory Committee at the VCCC was established in late 2015. It consists of 11 current members representing the VCCC Cancer Consumer Advisory Committee. The consumers are active participants of all nine program steering committees and working groups, providing the voice of the patient. The premises being that this will add value on many levels and improve results and progress in research. The goal of the Cancer Consumer Advisory Committee is consumer inclusion, embedding as business as usual across all VCCC Alliance activities. The key achievements for 2018 was a major consultation project to develop a consumer engagement action plan based on the National Framework for Consumer Involvement in Cancer Control, produced by Cancer Australia. After a study was conducted, it was determined the need for a standard approach that would influence and raise standard of consumer participation. A comprehensive toolkit will be launched in 2019 across the VCCC partners, enabling researchers and consumers to work effectively together. A similar program has been running since 2014 at the Walter and Eliza Hall Institute, demonstrating successful partnership between medical researchers and their consumers. The VCCC is at the start of an exciting journey where consumers will become the voice of the people to amplify consumer cancer research. The VCCC will offer new training programs for researchers on consumer inclusion, now essential for grant success. The first session, to be held in February, sold out in just 15 minutes. VCCC Research Conference in September also includes consumers on committee plus a public forum. For further information, make sure you're on the VCCC mailing list to get details of these and the launch of the Consumer Engagement Action Plan. You can find subscription forms on the website. Thank you.
Thank you, and I'll ask the speakers to come and join me at the front, if that's okay, to invite questions from the audience. I think we've got microphones roving around, and so if you do have a question, please say who you are and who the question's directed to. I'll step aside from the microphone, but while you've all got your thinking caps on, I might ask the first question, if I may, directed towards Claire. It's certainly something that we've been thinking heavily about at the VCCC with regards to best models of consumer engagement to support meaningful research. And one of the questions that I have, perhaps in NOMINATOR or one of your other studies, is in terms of your work with consumers, can you tell us about an example of where engagement with patients has modified perhaps the methodology you've used in a study, or the way that you've taken a particular study as a direct output of patient or consumer engagement you've had?
Well, certainly I think all my work with Rare Cancers Australia is an ongoing example, because we have a continual to and fro. Jonathan Granick also is my rare cancer buddy for my WeHi Stafford Fox rare cancer program, and so in that program, we have a whole range of different rare cancers that we study, and Jonathan will continually provide advice as to how rare cancer patients might want to engage. So their rare cancer patients have really different needs across different varieties of rare cancers, and so he will put himself in their shoes and perhaps, provide advice as to what stage in their rare cancer they might want to preferentially enter into research. So there, you have patients thinking from their perspective. So rare cancer programs are slightly different from clinical trials because they really are very flexible. I think in many other types of research, it is more clinical trial based. And so many of our clinical trial working groups have consumers involved in reviewing trial design. So their consumer input is equally important, because I think their consumers provide advice on trial design. And we have many good examples in my history is breast cancer and ovarian cancer, where consumers equally can influence the design of a trial and patient inclusion, or the way in which we might be affecting, perhaps the way patients have to attend clinics and so forth. They would be the main examples I have personally.
Thanks. I have a question for Katya and also Richard. Perhaps both of you could comment. Do you think in this age there is a strong consumer voice in setting research priorities in cancer, or do we still have a way to go? Where do you think we're at?
Yeah, most definitely. Consumers, particularly in the cancer area, can add lots of value. They can talk about their own personal experience. They can talk about the drug treatments. They can talk about the experience from beginning to end for them. I think they definitely have a lot to add value, and it's just a voice, quiet voice, that's getting louder and louder.
Yeah, I think we need to keep reminding researchers, and my favourite way of reminding you is that if you ever watch South Park, there are two people in South Park. There's one called Cartman, who's a bit tubby and a bit loud, but he's always got something to say. And there's another one called Token, who's an African-American in that cartoon who never speaks. And what we always say to researchers is, you choose whether you want Cartman or Token, because you've got a pretty fair idea which one we are, and it's important. I'm involved with another trial, and a health economist wanted to give terminal cancer patients a 90-page questionnaire around their economic status, and we tried to point out gently and then with a slightly stronger voice that that's insane, and it's also insane to give people of varying ethnicity, varying cultural exercise, an English-written, complicated document. Not everybody in this country reads and writes English perfectly, but they shouldn't be excluded. We can't continue to design a health system for Anglo-Celtic Australians. We're bigger and better than that. So, it makes a difference. We need to be noisy, and you need to be welcoming, is really how I would put it, because it will be valuable, I promise. Well, present company accepted.
Thanks, Richard. And who knew there'd be so many life lessons from Cartman? But Heather, did you have anything you wanted to add to that? Then, are there other questions?
Heather, my question's for you. I was wondering whether you had some advice for other consumers around the benefits of participating in clinical research, if there was something that you could say to them to encourage people to participate.
I'd encourage anybody to participate because you don't know what they're going to find. I mean, there could be a cure next week. I mean, so I would encourage anybody to join because, like in my case, I wasn't sure what my life, where it was going to go. And so when I was offered the opportunity, I just jumped at it, and I've been telling everybody about it ever since. So, yeah, I'm thrilled. Yeah.
Is there one final question from the audience? I think then on that note, there's one thing left to do, which is to put your hands together for a raft of such wonderful speakers.